speed up the reaction. See, we know already that in our body we have reactions, different kinds of reactions that occur in our body, right? All of those sum of reactions are also called metabolism. So this metabolism should be controlled by a specific catalyst, and the reason behind this control is so that it could be faster and faster, and we should get the product is much, okay, is it gain in different reactions. So that is the reason enzyme has a significant role in our body in different reactions. It may be catabolism reactions where molecules may break, and it may also anabolism reaction in which molecules will going to form. So this is a journal introduction about enzymes. Okay, enzymes are, again, they are catalysts. They will going to speed up the reaction. Okay, so if we see the enzymes nature, okay, so we observe that the majority of the enzymes are protein in nature. Okay, so they are globular proteins, means they are protein in nature. But there is an exception. Okay, there is an exception with one enzyme, and that is ribozyme. Okay, ribozyme is the enzyme which is not protein in nature. Okay, I said just now that the majority of enzymes are protein in nature, right? The globular protein. But there is an exception. There is an exception in which ribozyme is an enzyme which is not protein in nature. Okay, like if it is not protein in nature, so what is the nature of ribozyme? Now this is the next question. Okay, ribozymes are actually consist of RNA and ribosomes. Okay, so see, we are not talking about any protein here. We are saying that the ribozyme is made of RNA and protein, right? So that is the reason ribozymes are exceptional case in the field of all of the ribosomes, right? All of the enzymes. Okay, so like what is the example? Okay, what is the specific example of ribozyme? So the answer is peptidyl transferase. Okay, so this is the specific enzyme which is ribozyme in nature, peptidyl transferase, and it has a very vital role. Okay, and that vital role is the formation of proteins, and that is protein synthesis. Okay, protein synthesis. Okay, so if we see the diagram, okay, and if we want to see what exactly ribozymes looks like, right? So see, this is a diagram of ribozymes. It is made up of RNA. You can see these are some of the nucleotides, right? Nucleotides here. So these are ribonucleic acids, right? These are RNAs, and we know that RNA is one of the type of nucleotide, right? So we already know that we have two basic types of nucleic acids. One is RNA and the another is DNA. But in this particular case, we are focusing on RNA. Okay, so the next thing is about shape of enzymes, right? See, as we have already discussed that enzymes are protein in nature, majority of the enzymes are protein in nature. That's what we have discussed, right? So, if we see what is the exact structure of enzymes, they are complex as they are protein in nature. So, we have already the knowledge of the organization of proteins, like the level of organization of the proteins, like they are primary proteins and then secondary proteins and then tertiary proteins and then quaternary proteins. So in the same way, enzymes are also very complex. Okay, there will be with lots of folds. Okay, with different strains, there will be different strains, and there will be different um, we have different amino acids will be organized in a very complex form. Like see, here is a structure. Okay, this particular structure is complex structure. Okay, so this means like the enzymes will have such kind of structures. Okay, they will be like they may be tertiary, they may be quaternary structures. And if we see that what are these, right? What does exactly means about tertiary and quaternary? The word tertiary and quaternary specifically gives us the concept of different chains or different strains in that particular structure, right? Like we have alpha structures. Okay, we have uh, pleated sheets. Okay, so these are actually strains like alpha structures and then pleated sheets. Okay, so if we see what are the basic structure of alpha, like if I draw a diagram here, the alpha sheets diagram, see, this is pleated sheets and these are alpha sheets, right? So you can see here as well, right? These are strains, and these strains will be in a regular organization, and they will form quaternary and tertiary structure. Okay, so the next thing about enzyme, and this is the last portion of our lecture of today's lecture, and that is substrate and active site. See, if we talk about enzyme, there are other important parts or sites of enzyme, and these important parts are required for enzyme action. Okay, number one is we already know enzyme, and there is another terminology that is substrate and the active site. So we will see the concept of substrate and enzyme one by one. See, if we talk about enzyme, uh, enzyme, as we have already explained, that enzyme is a catalyst. Okay, enzyme is a catalyst, and that catalyst will going to speed up the reaction. So now the question is, what sort of reaction will happen in this case? Okay, so sorry, enzyme actually act on substrate. So the actual target of enzyme during the reaction is on substrate. Okay, so see, if we see the definition, we can see here like the substrate is the substance on which enzymes act, right? So see, this is the diagram. Okay, so in this diagram you can see pink, you can see the structures in pink color, and these are actually substrates. Okay, so these are the particular substances on which this enzyme will going to act. Okay, and this particular substance will be substrate. Okay, so like you can say these are reactants. Okay, reactants in a particular reaction where enzyme is just taking part in like making the reaction proper and specifically to speed up the reaction. Okay, and for that reason enzyme is going to act on this substrate. Okay, so see, the reactant which to be converted into product, so this substrate is going to convert into another form, and that is product. See, this pink like structure which we have called is substrate. Okay, so this substrate is eventually become a product. So this has been converted into product, right? So that's what this line explains here, right? This line is explaining that the reactant which is to be converted into product. So it means the substrate is going to convert it into the product, and this is happening because of enzyme, because enzyme is acting on substrate. Okay, so the particular place where substrate is attached on enzyme is called active site. Okay, so see, uh, the substrate will have a specific site, a specific area within that enzyme, because enzyme is a globular structure, you can see here, right? This particular enzyme, but there will be a specific region for specific substrate. And see, the substrate shape will going to define that particular active site. So that is the reason substrate is specific, okay, and it has a particular site within that enzyme. That is why that site is called active site. See, this is the area actually. That is the reason it is called active site. Okay, but okay. So the specialized site, okay, like see, this is the particular area where there will be two processes, okay, there will be two processes. These processes will go into perform, and that is catabolism and anabolism, okay. So these are the reactions which will going to perform in that particular area, and that particular area is called active site, okay. So if you go into the depth of the nature of active site, like what exactly active site is and what is the nature, how it this active site actually form. So the answer of all of these questions is the active site is made up of charged amino acids. See, this active site is made of charged amino acids, right? Charged amino acids, okay. Three-dimensional cavity, like it is three-dimensional, okay, that the particular area is three-dimensional, and the sort of enzymes here are, charged amino acids. So that is the reason it is attached, active site, like active site has the capability of the attachment of the substrate. The reason is here are charged amino acid. That is the reason. So this is actually mechanism of enzyme catalysis, where what is happening, the enzymes are uh, the enzymes are doing their work by increasing the speed of a reaction, okay, and this particular enzyme is given a particular place for the substrate in that place, and that particular site is called active site, okay. So the last segment, okay, the last segment is about types of active sites. See, we are actually focusing on the nature of active site, as we have said that the active site will contain charged amino acid, okay. So that is the reason the substrate is going to attach, okay, on the particular area, and that is active site, okay. So see, like we have two categories of active sites. One is called okay, where is the okay, one is called binding site, okay, and the another one is called catalytic site, okay. One is called binding site and the another one is called catalytic site. See, in this diagram, in this particular diagram, let's find out where is binding site and where is catalytic site. See here, catalytic site, and there will be binding site. So see, this is the binding region and this is the catalytic region, okay. So uh, we will see what is the function of this particular catalytic site. The main function of catalytic site is to recognize the substrate, okay. So we have already discussed that substrate is going to attach on the particular active site, but there should be prior preparation, and that prior preparation is being made by this binding site, because this binding site is going to recognize the relevant substrate, okay. The relevant substrate means the particular substrate which will match the active site. That is also the important part, okay, that this should be very essential for this enzyme mechanism, that the enzyme and substrate should be matched. That is the reason we say that the substrates are specific, because the active sites are designed according to the shape of substrate. That is the reason, okay. So this is very easy, okay. Binding site, the word is actually giving us the clue that this will going to bind the substrate, and apart from binding, we should also understand that this will going to recognize the upcoming substrate, okay. So the next thing is about catalytic site, okay. The catalytic site, the word is actually giving us the clue already, right? Catalytic means catalysis, okay. So this will be the particular place where the reaction will genuinely occur, right? So see, if we go through with the definition, this definition also tells the same thing to us, right? Catalysis of substrate and the reaction will complete. So this is the particular site where the reaction will going to complete, and if we dig into the concept of what sort of reaction, as we have already know this, that the substrate is the participant here, right? So in this case, the substrate will going to convert into the product. And this concept has already been cleared with previous slides, as we have discussed that the substrate is going to convert. Okay, substrate is the particular reactant that will going to convert. Okay, so this is the all over concept about binding site and catalytic site. Okay, so this line, as I have already described, that the active sites are designed according to the substrate nature. Okay, see, if this is the substrate, you can see that it has the particular shape, you can see, right? So according to the particular shape, there will be the active site, and this active site will going to attach this substrate. Okay, and for this purpose there are two types of binding sites. One is called sorry, active site. One is called binding site, and the another is called catalytic site. Okay, so these were the topics of today's lecture. Thank you so much for watching the whole video. Do like it, and subscribe it as well. Thank you so much. Thank you.